Thank you very much, Luke and Lizzie, for your excellent presentations and getting us back on track. I'd now like to ask both of the grantees from last year's awards to join us on the podium. And following the theme of what you've heard, the first speaker is going to be Lipika, who will then take that a step forward and talk about FGFR emergence of resistance before we hand over to Sumira. So when you finish, please remain on the podium, and then we'll go to questions.
So thank you very much to the Cholangiocarcinoma Foundation for supporting our research and for calling me here to speak about our work today. I also want to thank Aram and Juan. I'm so sorry. He was just up here. Aram and Juan for inviting me to speak today about our work. We have been really encouraged by the response to BGJ398 in patients who have FGFR aberrations and cholangiocarcinoma. People are having incredible reduction of their tumor in second, third, and fourth line areas. And we're finding that while people are having excellent responses, and some people are having them for a prolonged period of time, some people are having responses only for a short period of time, like 4 to 6 months. And as a clinician, I find this really disheartening because I want, obviously, for my patients to have these treatments for 1, 2, 3 years. And so it behooves us as clinicians and as scientists to try to understand why people are not responding for longer so we can figure out ways to prolong those responses. Now, Aram and Juan did a wonderful job of putting Lizzie and I in touch before this presentation. And so I have 0 background slides on FGFR. So you won't have to hear any of that stuff all over again. But I do have 1 background slide on oncogene addiction. Sorry, where's the clicker? So as they say, cancer is 100 different diseases masquerading as one. And so I think one of the biggest accomplishments over the last couple of decades in translational oncology is being able to divide cancer into 100 different diseases. And in some cases, trying to figure out what is the one gene that is turning on someone's cancer so we can give them a drug that turns that off. And there are several examples of this in oncology. The definition of oncogene addiction, which is a term that was coined by Bernard Weinstein in 2000, is this curious observation that a tumor cell that has a plethora of genetic aberrations can seemingly exhibit dependence on a single oncogenic pathway or protein for sustained proliferation and survival. So the earliest example of this was BCR-ABL. BCR-ABL is a fusion that is seen in chronic myeloid leukemia. The Philadelphia chromosome was discovered in 1960 and was characterized by Nolan Hungerford. And then in 1986, it's when they figured out that it was a translocation between chromosome 9 and chromosome 22, and it led to this transcript called BCR-ABL. In 1996, they found that the drug imatinib showed promise in preclinical models, and very quickly this moved into a phase 1 trial with Brian Drucker in 1998, and then it was very quickly approved by the FDA in 2001. But as you can see, there was a 40-year gap between when we discovered the Philadelphia chromosome and when we were able to get imatinib FDA approved in CML. Now, there are multiple examples. I just chose 3 solid tumors to give an example. One is EML4 ALK-positive lung cancer. The other one is EGFR-positive lung cancer. And the last is c-KIT or PDGFR alpha-positive GIST. So these 3 cancers, I think, are very interesting because we have a tyrosine kinase inhibitor that's a first-generation inhibitor that has been successful in all of these different cancers. So ALK is ALK-positive lung cancer. That's about 3 to 5% of non-small cell lung cancer. EGFR, it's about 25%. And c-KIT, it's actually, c-KIT mutations are seen in about 90% of GIST. And of the remaining patients, about 35% have mutations in PDGFR alpha. And so we have drugs such as crizotinib, gefitinib, erlotinib, imatinib to treat all of these different diseases. But what we see is that in all of these different diseases, when you give a tyrosine kinase inhibitor, people develop gatekeeper mutations. So how did we come up with the idea for this project? We basically stood on the shoulders of giants because we know with tyrosine kinase inhibition, people develop these mechanisms of resistance. So having this in mind is how we developed the idea for this project. And something interesting about FGFR is that it is found in 10 to 20% of intrahepatic cholangiocarcinoma, but it's also found in a lot of different cancers. There's a paper from Greenman in 2007 in Nature which showed that of all the different tyrosine kinases, FGFR is the most frequently mutated in all of cancer. And then there was another paper by Helston in 2016 in Clinical Cancer Research, and they looked at almost 5,000 different tumors, and they found that there was some genetic alteration in FGFR in about 7% of tumors. So I think this is actually a very critical moment in history because FGFR at some level is the new ALK EGFR c-KIT, and it's present in so many different cancers. And we have seen, as Lizzie presented a slide of all these different FGFR inhibitors that are now hitting the clinic, they're hitting the clinic in a lot of different cancers. So this is the moment that we need to be thinking about resistance so we can prolong responses for our patients. So this is the roadmap for my presentation. I'm going to talk about the 3 patients that were a part of this project. I'll talk about the identification of the mechanisms of resistance to this drug, BGJ398, and then I'll talk about how we functionally validated these resistance mechanisms. So how are these patients identified for this study? So at MGH, we have an internal assay that we use to detect fusions. It's an RNA sequencing assay. The one benefit of RNA sequencing is that for our assay in particular, it's agnostic to the fusion partner. So the way it works is we have from formalin fresh from FFPE tissue, we basically extract the RNA, and then you can't do PCR off RNA, obviously, so you do complementary DNA, and then we develop primers that are targeted primers against the FGFR2 gene, and then we have primers against the universal adapter at the other end, and then you sequence it, and it doesn't matter what the fusion partner is, because when you do the alignment, you can figure out what that protein is. So in this way, the assay is able to detect fusions that have not yet been reported in the literature. And so we were able to find about 28% of our patients had FGFR2 fusions. This is obviously an enriched population because a lot of people that we're testing were people who were ready for second or third line therapy, so maybe they had indolent diseases. Obviously, if we had young female patients with cholangiocarcinoma, we're also more likely to run this assay. So I wouldn't say that the prevalence is as high as 28%, but this is what we found with our assay. And then we found all these different fusion partners. 4 of the patients enrolled on the trial, and 3 of them had progressed by the time of the paper was written. So this is the waterfall plot, and our 3 patients were somewhere around here. They had a 28%, 37%, and 50% reduction. And then this is a swimmer's plot where we see some people who responded for a long time and some people who responded for a short time. And these are our patients around 3.6, 5.6, and 7.4 months is how long they responded. So I'll start with the clinical history of the 3 patients. The first patient was a 59-year-old woman with unresectable intrahepatic cholangiocarcinoma. She had a 14-centimeter tumor, and she had that spanned her left and right lobe and a couple of little satellites on the left. She got 10 months of gemcitabine and cisplatin, and we routinely do genetic testing up front so that we're ready for plan B when a patient progresses. And we knew ahead of time that she had an FGFR2 alteration. It was a fusion with the zinc finger MYM protein 4. And this was a fusion between exon 17 of FGFR2 and exon 17 of FGFR2 and at exon 10 of ZMYM4. And she went on to BGJ immediately after GemCis. The second patient was a 47-year-old woman with recurrent metastatic intrahepatic cholangio. She actually had a tumor for a couple of years in her liver that we were biopsying and following, and we weren't able to diagnose cancer. And then eventually when it grew a little bit bigger, we biopsied it. It was intrahepatic cholangio. She went to surgery. An 11-centimeter T2N0 tumor was removed. She got 6 months of adjuvant therapy, and then 5 months later, she unfortunately had a spot in her liver. She underwent some liver-directed therapies, such as ablation and radioembolization, but her tumor progressed through these. And then we also, for her, in advance, knew that she had a fusion between FGFR2 and the optineurin protein, optineurin gene, OPTN, at exon 17 and 5. And she went on to BGJ. And the third patient was a 43-year-old man who was from Europe and used to fly to Boston in order to be able to get this drug which speaks to the commitment and testament of how strong our patients are. And he had primary metastatic intrahepatic cholangio. He had tumors in his lung and his lymph nodes. And he had palliative chemotherapy, and then when he progressed, he went on to onto BGJ398, and he had an FGFR2 BiC1 fusion at exon 17 and exon 3, respectively. And so these are the radiological outcomes of our patients. As we talked about, they had a 50%, 28%, and 37% reduction. And then all of them, unfortunately, had progression. What's interesting about all three patients is they all had a mixed response. So there were some tumors that were shrinking or stable while others were growing at the time of progression. And in some cases, people developed a new lesion. And so we also looked at the CA19-9 and resist trends. So the pink boxes are the time when the patient was on BGJ398. And you can see for the first patient, the CA19-9 is the green line and the tumor volume is the blue line. And you can see that while the patient was on treatment, both the tumor volume and the CA19-9 went down. And then as she started to progress, these both went up. Similar for patient 2. And then for patient 3, his CA19-9, as is true in about 10 to 20% of our patients with cholangiocarcinoma, was not informative, but you can see his tumor volume went down with treatment. So now I'll talk about how we identified mechanisms of resistance. So as Andrew talked about, we have an acquisition protocol at Mass General on how we collect tissue and plasma samples. So basically, we wanted to look at baseline, on treatment, and post-progression tissue and blood so we can understand what was emerging on treatment. What we found was on post-progression biopsies of these patients, we found the appearance of the FGFR kinase domain mutations. 2 of them had biopsies, 1 did not. So we found kinase domain mutations in 2 of them on biopsy, but in all 3 of them on circulating tumor analysis. And we have a rapid autopsy program at Mass General where people who have interesting genetics and have been on targeted therapy or are interesting for any other reason, our patients have been really generous and have donated their tissue to science. So we rapidly do an autopsy, ideally within 6 hours of passing. And in one patient, we were able to learn a lot about FGFR resistance because of this generosity. So the first patient, Andrew also showed these slides. The patient at baseline had no mutations in FGFR2 in her tissue or her blood. And then at progression, she had 2 in the tissue and 5 in the blood. Notably, the red one is the gatekeeper mutation. The second patient had, again, no mutations at baseline in her biopsy or in her blood. And then at progression, she also had 5 on circulating tumor DNA analysis, and she had only 1 in the biopsy tissue. This is a patient whose family generously donated her body to science, and we were able to try to understand better why we found 5 in the blood and only 1 in the tissue. And I'll show that autopsy data in a couple of slides. The third patient, he was on treatment the longest, and he only emerged 1 mutation, which was the gatekeeper mutation, and he did not have a post-progression tumor biopsy. So we also wanted to see the emergence of these different clones over time. So this is work that we did with Dr. Bardelli in Italy and his team and Ryan Corcoran. And so basically we used ddPCR to look at levels of the individual resistant FGFR2 clones over time. We checked at baseline 1 to 4 months and at progression. The black line is a founder mutation that basically helps us see what was happening with the tumor volume over time. The black line in the first one is an ARID2 mutation, and you can see that in the beginning it was high, while she was on treatment it went down, and as she started to progress it went back up again. And you can see that the FGFR2 mutations were right at the baseline. They were undetectable initially, and then just before month 4 they started to increase, and she actually came off treatment at month 4, and you can see over time they continued to go up. And same thing with patient 2 and 3. You can see the black line shows that the mutations went down initially in terms of the founder mutation, but all of the different resistance mutations, which were not present at baseline, emerged at the time of progression. So this is the autopsy that we did on that second patient. This was in collaboration with Shup Saha and Dan Urich, who is the head of our autopsy program, and Jim Stone, who is the director of the anatomic pathology unit at Mass General. Everyone came in at the middle of the night, and we performed this autopsy around 1 a.m. And it was really an instructive moment for me to understand how our scans sometimes underestimate exactly how much tumor people have in their bodies, because this scan was done about 3 weeks before this patient passed away. And as you can see in the actual autopsy, her entire liver was pretty much taken up by tumor. And so there were greater than 20 different metastases, but we sampled about 15 of them in order to try to understand where these resistance mutations were coming from. So this is a heat map of the various mutations. So the horizontal lines are all individual mutations. The vertical lines, the columns, those all represent one tissue sample. And so we sampled 15 different lesions, and 12 of them had at least 10% tumor cellularity, and we sent them to Foundation Medicine for targeted deep sequencing of 315 genes. And I want to call your attention to the bottom 3 rows. These are the FGFR2 mutations. So as you can see, in the 12 different lesions that we looked at, most of them actually did not harbor FGFR2 mutations, but 4 of them did. The 4 that did, each of them only had one FGFR2 mutation, except for one lesion, liver met number 5, which had 2 different mutations. So one question is, why did only 4 of the lesions have FGFR2 mutations when this patient progressed on therapy and developed resistance? So one, as I mentioned, she had a mixed response. So some lesions were still responding when she came off, and some lesions were resistant. But also, this autopsy was done 3 months after she came off treatment from BGJ. So it's possible that in some of the lesions that were resistant, there was some growth of the sensitive clones that overtook the resistant clones when there wasn't the selective pressure of BGJ any longer. And we see that right here, where there are these 2 lesions. You know, we tried to basically correlate with the radiology which lesions were resistant and which lesions were responsive. And we have 2 lesions that were resistant that did not have any FGFR2 mutation. And again, this could be because the sensitive clones outgrew them. Another question we wanted to ask was, did these mutations emerge in fusion positive or fusion negative cells? And so with Foundation Medicine, we were able to detect the FGFR2 fusion in 4 of the 12 metastases, but we had a suspicion that all 12 metastases actually had that retained the fusion. So this is some work by Leah Liu in Nabil's lab, and using RT-PCR, we were able to find that the fusion was actually present in all of the different lesions, confirming that these mutations did in fact emerge in fusion-positive cells. So that previous slide showed intertumoral heterogeneity, how different clones arose in different metastases. This is a slide showing intratumoral heterogeneity. Again, speaking to Dr. Gore's question, basically we took one of the lesions and we cut it up into 8 different pieces and sent it to Foundation for sequencing. And what we found was 3 different loss of function mutations in P10. Now, the significance of this is unknown, but there must have been some sort of selective advantage for these different clones to develop a mutation in P10. And there is some preclinical data in FGFR2 amplified gastric cell lines showing that P10 knockdown may be a resistance mechanism to FGFR inhibition. So this is just hypothesis generating, and further work certainly needs to be done. So we were not the first people to identify these mutations. These mutations were predicted in preclinical models using a BaF3 system that was dependent on FGFR2. This is work by Byron and colleagues where they exposed a BaF3 system to increasing doses of dovitinib, and they found 14 different mutations in FGFR2 in the kinase domain, including all 6 of the different mutations that we saw. And they also looked at what was the mechanism of how these different mutations caused resistance to FGFR2 inhibitors, such as dovitinib. So as we talked about, all 3 patients had the gatekeeper mutation, and as I showed on the first slide of oncogene addiction, gatekeeper mutations are one of the most common causes of resistance to any of these tyrosine kinase inhibitors. And so with some in silico modeling with the help of Pascal Foret at Novartis, we were able to show that when you have a gatekeeper mutation, it causes steric clash with BGJ because this residue basically interacts with the dichlorodimethoxyphenyl ring of BGJ. And when you have another amino acid, so F is phenylalanine, it has a bulkier side chain which interacts with BGJ and doesn't allow BGJ to bind to the ATP binding pocket. The Byron team also looked at the other mutations and why they cause resistance, and I'll just, basically all of them destabilize the inactive form of the FGFR kinase domain to which BGJ binds. And I'll just highlight the triad N549, E565, and K641. They form the molecular brake, which stabilizes the FGFR kinase domain in the inactive form. But when you have a mutant in one of these different molecular brake residues, it basically disengages the molecular brake and allows the FGFR kinase to be active. So we have all these different mutations that we've predicted. What can we do about them? So Nathaniel Gray's group basically tried to develop an inhibitor that can overcome the gatekeeper mutation. So he was able to develop this drug that he called FIIN2, which is an irreversible inhibitor that covalently binds to the cysteine residue that is present in the glycine-rich loop of the binding domain. And because of this, it actually allows the drug to bind in the DFG out mode, and it allows, because the drug is actually quite flexible, it's able to slip right in beyond the gatekeeper and actually bind even in the presence of a gatekeeper mutation. And so you can see the EC50 is actually in this case, where for BGJ, for the gatekeeper mutation, the EC50 was 1500. You can see for FIN2, it was 58. And for the other gatekeeper also, it was 3,000 compared to 100. So this drug, the whole point of this study was to develop a structure and give basically pharmaceutical companies some guidance on a blueprint on drugs to make irreversible inhibitors that can be used for patients to overcome gatekeeper mutations after the development of these on prior inhibitors, or for irreversible inhibitors to be used up front so we don't emerge these gatekeeper mutations. So lastly, I'll talk about the functional validation of the resistance mechanisms and the drug testing that we did. So the two questions we were asking with the functional validation were, are all resistance mutations created equal? Are some of them more resistant to BGJ than others? And secondly, what FGFR inhibitors can we give to our patients after they develop resistance on a drug such as BGJ398? So we were working with a TEL-FGFR3 fusion system that Novartis had, actually. And so we wanted to see if FGFR2 and FGFR3 are homologous, so that any data that we found preclinically with FGFR3 could be applicable to our patients with FGFR2. And indeed, what you can see with this alignment diagram is that FGFR2 and FGFR3 are 89% homologous. So we felt that the data could be used for FGFR2 as well. So we did a mutagenesis screen with BGJ in the TEL-FGFR3 BaF3 system, and we gave increasing concentrations of BGJ398, 100, 200, 400, 800, and 1600 nanomolar. And what you can see is at low doses of BGJ, in the screens, 5 different resistant clones emerged, and these were all corresponding mutations to the mutations that we saw in our patients. But when you give high doses of BGJ, only 1 clone emerges, but 1 clone still does emerge, and that's the gatekeeper mutation. And again, this is another graphical way of showing which mutations are more resistant than others. So the red V55M and V564M, this was the most resistant mutation. The IC50 was 1,000-fold compared to wild type, and the other mutations were a little bit less resistant, but still resistant nonetheless. So the last part of our work was to do a drug sensitivity screen. So we basically took these resistant clones and used 5 different FGFR inhibitors that are present in clinical trials right now, and also this drug by Nathaniel Gray, FIIN2. And what did we find? We found that this compound by Lilly was the most potent drug in being able to overcome resistance. So you can see the IC50s for the gatekeeper mutation is 0.32. And and that was much lower than for BGJ, which you can see is 600. So these kind of preclinical studies give us insight on what kind of inhibitors we could be using beyond progression in patients who have been previously treated with FGFR inhibitors. So in summary, in terms of what kind of future directions we can take, FGFR alterations are present in 10 to 20% of intrahepatic cholangiomas and also present in multiple tumor types. Clinically acquired resistance to BGJ occurs through mutation of the FGFR2 kinase domain. And I hope I've convinced you with our presentation that it's really important to be able to get baseline blood and tissue on patients and then during treatment and progression so we can understand why people are developing resistance and which are the resistant clones that are developing early and how that can inform our strategies on thinking about which drugs to use and what kind of combinations to use. The bad news from our study was we found this concept of intratumoral heterogeneity where there were patients who were developing polyclonal resistance. They were developing 5 different mutations. And as a clinician, that's scary because we're like, how are we going to overcome all of these different resistance mechanisms? But the good news is that we do have drugs that can potentially overcome these resistance mechanisms. And there are lots of different FGFR inhibitors right now in the clinic. And we don't know which one is best, but I think with this preclinical modeling, it helps us give some insight into which ones might be potent against the resistant clones. And my vision for the future for FGFR is that we can turn FGFR positive cancers such as cholangio into ALK positive lung cancer and EGFR positive lung cancer where we can have multiple lines of treatment that we can give to patients, first generation, second generation, third generation inhibitors, so we can turn this disease more into a chronic disease rather than into something that takes people quickly. So thank you very much to everybody. I just wanted to also acknowledge a few people. This was a huge collaborative effort with 32 authors on the paper and multiple labs involved. And I want to echo what Nabil said about community matters. Most importantly, our patients and their families who so generously donated a little bit of tumor tissue when they were getting biopsies, and of course, donated tissue. Sorry, I don't know how to go backwards. If we could go back one slide, that would be great. Donated tissue for autopsy also, which was really helpful. I wanted to acknowledge the Bardisi lab because they teach me every day how much faster we can come up with answers when we have clinicians and scientists work together. It's been a wonderful collaboration over the last several years. I wanted to thank Ryan Corcoran and Dan Urich for their scientific input throughout this project. And then most of all, I wanted to thank Andrew. When I was a fellow 5 years ago, I came to Andrew because I really wanted to work with him because the advice I got early in my career was follow people. And at Harvard, when we go through fellowship, we work at Dana-Farber and Mass General for 6 months each, and you have to, at the end of the day, pick a disease center and pick someone you want to work with. And I had worked with Andrew for a couple of months when I was at MGH, and I was like, I'm working with Andrew. That's how I chose going into a career in hepatobiliary cancers. And he has been an incredible mentor over the last several years, and I'm standing up here on this podium in large part because of him, because he gives me a wonderful balance of autonomy and guidance to do all the work that we do. So thank you, Andrew, for all your support for the last several years. Thank you very much.